everybody to SynBioBeta Live. What a great time it is to be back in the hot seat. Today we have a fantastic event sponsored by Riffin, and very shortly we'll be joined by Tim Gardner, the CEO of Riffin, plus a veritable banquet of panelists today to talk about digital infrastructure, and in particular how you can manage a lot of people working from home, a lot of people still working from home, and a lot of people needing to do distributed R&D. So some very exciting work that's going on in the lab and also at the bench as well. And we're going to be talking about some of the issues that you have around getting that data into a format where you can run all sorts of machine learning tools and all sorts of other tools that you can use to get more from your data. If you're just joining us, welcome. This is SynBioBeta Live, and my name is John Cumbers. I'm the founder and the CEO of SynBioBeta. And we're broadcasting live today from California, and we're joined by a veritable banquet of panelists. We're going to be going through and speaking to all of the different panelists about what they're working on, how they're using tools such as Riffin Nexus, which just relaunched a couple of weeks ago. And if you are interested in a demo of the platform, then at the end of today's session, you can go and get a demo from the Riffin team who is standing by. We'll also be taking your Q&A right here. So we're going to dive in, and before I do, I want to remind you that SynBioBeta Digital is coming up on September 1 through 3, and we have over 100 speakers who are going to be speaking at it this year, some very exciting topics. And also, if you haven't signed up to our newsletter, then I encourage you to go to synbiobeta.com and sign up to the newsletter. So Riffin Nexus is the world's first process data system, a cloud-based data system that uses R&D and manufacturing processes as the organizing principle for data storage, analysis, and user interface design. And really, as technologies are becoming cheaper and increasingly more precise, many pharmaceutical products are taking longer and longer to reach the market than ever before. And recent global trends have challenged our ability to perform scientific pursuits in the same old ways. The need for transformation in how we work is more clear than ever. And SynBioBeta always brings together leaders in the field of synthetic biology to discuss how we can reimagine R&D, digitization, and biomanufacturing to address some of the urgent consumer and industrial trends and the ever-changing, accelerating world. So today we're going to be talking about making data more, how to make more discoveries with less effort, the factory lab and how principles of manufacturing and biomanufacturing, quality and data analytics assist R&D effectiveness without inhibiting innovation. We're going to be talking about distributed research, how contract research and digital collaboration technologies are changing the way we do science, and reduction to practice, case studies in successful digitization. If you are new to SynBioBeta, then we'd love to know where you're dialing in from, and there will be a dialogue going in the chat. So if you want to type into the box and tell everybody where you're from and where you're dialing to, don't just submit it to all panelists, but submit it to everybody. And there'll be an opportunity, if you have questions, you can type them into the Q&A box and you can ask the panelists live any questions that you might have if you want to join in the discussion. So as I said, we have 5 panelists today. The first person we're going to be speaking from is Tim Gardner. He's the founder and the CEO of Riffin. The second is Lauren Perelman, who is the Vice President of Scientific Consulting at Riffin, and Lauren's going to be giving the keynote very shortly. Next, we have Ken Finley, who's the Principal Biotechnologist at Cargill. Then we have Elizabeth Irons. She's the CEO of Science Exchange. And finally, we have Jessica Louie, who is the data science lead at Join Bio. And Join Bio is a partnership between Ginkgo and Leaps by Bayer, looking at crops and agriculture. So as I said, following the panel, we're going to be having a demo where you can look under the hood and see how the Riffin platform works. And we're going to be able to continue the conversation here if you have specific questions for Lauren. We're going to be taking Lauren's keynote very shortly, and then we're going to be having each of the panelists talk a little bit about what they do. And then we're going to be having a moderated Q&A. And as I said, if you've got any questions, then please put them into the box and we'll take them as we go. And you can also raise your hand if you have a question, type it in the box, and we can call on you live. Tim Gardner is a close friend of mine. He's the founder and the CEO of Riffin. He was previously Vice President of Research and Development at Amyris, where he led the engineering of yeast strains and process technology for large-scale biomanufacturing of renewable chemicals. Tim, welcome to SynBioBeta Live.
Thanks, John. Good to be here.
Let's start from the beginning. Let's talk about some of the frustrations that you had in the lab and then why you started Riffin.
Oh, where to start? I've always had a foot both in the lab and in computation. And I think one of the core frustrations to me, sort of the frustration of frustrations, is the artificial segregation of computation from experimentation, as if they were two different things. And the real magic in science, just as it was in the case of 20th century physics, is when you marry computation and experimentation together. But to do that, you need to use data to inform theory and models. And you can't do that if data is of poor quality, because it's just garbage in, garbage out, and you end up chasing down completely wrong ideas. And that wastes a lot of time. So the frustrations of that dichotomy between experimentation and computation leads to a poor treatment of data quality, which then leads to almost self-fulfilling prophecy. They become separated because one doesn't inform the other.
We talked together and we wrote up a piece for Forbes on some of the problems that the biopharma industry in particular has with these challenges. And we talked about Eroom's law. What is Eroom's law, and what are some of the other challenges that we identified from that piece? And maybe we can type that piece into the chat box for anybody who didn't see it.
Yes, a great question. So Eroom's law is Moore's law backwards. Moore's law is that the power of computation, the speed and scale, the speed of it doubles every 18 months in silicon chip manufacturing. At least it used to. And Eroom's law is that the efficiency of biopharmaceutical research drops in half every 18 months, meaning it's that much worse every 18 months. And that's really held up. It's really true. There's publications on this. The foundation, sort of the principle of why Riffin exists, and sort of really our mission in science, is to reverse that trend, because we believe that it's not a consequence of biological complexity, which is often the excuse. Oh, biology is complicated, that's why it's getting harder. It's actually consequence of misuse of information and data. We produce data that's not usable. We make bad decisions. And as a result, we shoot ourselves in the foot, making our jobs a lot harder in science. And if you don't believe that, all you have to do is look at the speed with which we've developed vaccines for COVID under extreme global duress. We have vaccines that are in phase 3 trials now after like three months of development. At the start of those 3 months, everyone said it's impossible to have a vaccine any sooner than 18 months. Impossible. And all of that was based on the history of how long it takes to do drug discovery. But most of that time is not inherent to the science. It's inherent to our human structures and organizations. And so if you put your mind to it, the science will give way to extraordinary speed. And I think we can get there if we develop the right tools, technologies, and behaviors as a society and as a scientific endeavor.
Fantastic. And we'll come on in a second about how the Riffin Nexus platform enables that when we hear from Lauren. There was another industry about 70 years ago that was mired in high costs and failing products. That was the auto industry. What happened with the auto industry, and what lessons are there that we can learn from that?
Well, between 1950s, 60s, and 1980s, the Japanese adopted total quality methods, and they quadrupled their productivity. Quadrupled. The US industry kept doing things the same old way, and they got no increase in productivity. The consequence of that is the Japanese auto industry came to dominate the global auto industry. So the short lesson is quality leads to productivity, and productivity leads to, well, winning. And so it's the same in science. We learned that. Lauren learned that. I learned that. Many people learned that in our work in industrial biotech and synthetic biology, because we actually applied those principles, and we doubled and tripled and quadrupled the productivity of our research and development organizations.
I'm a big fan of Ray Dalio, and he has a fantastic video on the web, about a 30-minute video, kind of economics 101. And I was actually studying it lately. And productivity is the only thing that is it's the main driver, not the only thing. It's the main driver of economic growth is productivity. So definitely fantastic to get that example from another industry. I was struck by your comment, make greater data and great products will flow. Can you elaborate on what you meant by that?
Yeah, and I think maybe I'll use this as a transition into introducing Lauren, because there's a great story about this. So Lauren, who's about to give a presentation on his work in Riffin, we met maybe almost a decade ago, and he walked me into a big freezer that was used to store samples from purification process development and method development. And this room was full of bottles, all labeled, all different sizes and samples, all labeled with various hand labeling and safety information. And then he pulled a few of them out and he said, watch this. So he grabs a couple, he writes down the IDs, and he goes to the data system and types in the identifiers of a few of these bottles. And all of them get pulled up, and all of them have no information. Not only they have no information about what's in those, but there's, of course, zero information of how they got there. How were they processed? How were they created? Where did they come from? What were all the factors that led to the properties of those materials? And we kind of realized that, when we go into the lab, we think we're making materials. Oh, we're making this substance. Isn't it great? It's pure and it's clean. But we're not, because if we made the perfect substance and there's no information about how we made it, how can we ever do it again? We can't. So what we're actually synthesizing in R&D is data. We're synthesizing the data on the outcomes, but also the processes by which we generated those outcomes. And if we don't capture those processes with the same adulation that we capture our results, then we've gone nowhere. And that really is the core of what drives Lauren and I in our current research and current efforts.
Fantastic. Well, Lauren, I think over to you then. Lauren is Vice President of Scientific Solutions at Riffin, focused on providing customers with the best experience that they can, implementation, training, and utilization of the Riffin Nexus platform. And prior to Riffin, he was Director of Quality, Analytics, and Manufacturing at Bolt Threads. Lauren, welcome to SynBioBeta Live.
Hey, John. Thank you for having me today.
Absolutely. And I think you have some slides that you're going to share with everybody. Is that right?
Yep.
So if you want to go ahead and bring those up. And then, as we said at the beginning, this is a live event. And if you have questions, you can raise your hand. You can type them into the box and we'll get through as many as we can.
All right. Can you see my slide?
I can. Everything looking good.
Lovely. All right. So I'm going to build on today's theme, which was WFH to WTF to WFH. And I'm going to answer the question, what the F is a digital backbone? Because we're talking heavily about this data management and structuring data to make everything easier for all of us. So let's dive in. I would be remiss if I don't first talk a little bit about what we're trying to do at Riffin. And it's, I say with tongue in cheek, a humble mission to make science work better. And what that means for us, building off what Tim was saying, is it's delivering shorter, more predictable scientific product development, those cycles to global biopharma and biotech. We're really focused in, the pharmaceutical, synthetic biology, and biotech spaces. And so we could dig into what that means here. First, let's talk a little bit about the state of scientific discovery. That Eroom's law says that R&D is yielding less return on investment. And so, for example, you could see on this graphic here on the right that over the last 40 years, we've seen more than a 10x increase in the R&D cost per drug in millions. And now we're at about $2.6 billion to get a drug to market. And that's interesting because, sort of in contrast to that, we have the most incredible tools that we've ever had in the scientific enterprise. There's just incredible sophistication in our work streams. In all of these different places like fermentation and downstream processing and materials processing. It's incredible what you see today, both in terms of what can be done at the bench, the different molecular biology techniques that we have, the computational techniques we have. And yet teams are often still siloed and somewhat inefficient in that holistic data picture. So, you've got data, but each team is sort of looking at their part of it as an independent project all on its own. And so, there's still so many opportunities to get more out of what we're doing, to gain more efficiency from our workflow and data flow execution, and more opportunity, especially to employ powerful algorithms for machine learning, to really get in there and understand more about our data. And core to this is this data wrangling problem. And I'm sure for all the data scientists on the line, this is near and dear to your heart. Even today in 2020, you'd think this is a no-brainer, right? Let's structure our data. Let's make it easier for ourselves to get work done. And yet, time after time after time, when we talk to our customers, this is one of the core problems that they're dealing with. And so you see this this quote on the left that really it really highlights just how bad it can be. This is not uncommon. They're saying it's taking up to 2 to 3 months to assemble a single batch of data, which is crazy because isn't time money and data money in a pharmaceutical space? And that's just lost opportunity. And that's that 80% problem that we're all familiar with. So I guess that begs the question, what is and is not perhaps a digital backbone? It is a system that captures process and data information, and it manages those process versions across multiple scales and the history of those processes. Ideally, it conforms to FAIR data standards. It should enable more rapid and deeper discoveries and holistic integration of heterogeneous data. So that problem we saw on the last slide should be eliminated if we have a strong digital backbone. What it is not, and this is a problem for a lot of companies, it's not simply paper on glass. It's not moving the lab notebook into an electronic lab notebook, but then leaving each person's work on an isolated island. And it's certainly not just capturing information in a digital format. So, procuring beautiful Excel files is lovely, but in isolation, they're not really doing much with one another. And I think, it's like every decade there are certain buzzwords, and digital transformation is one of those buzz terms right now. And so for certain executives, I think it ends up being a checkbox activity. And so that's not really helpful if you don't enter this, the goal of a digital backbone with a lot of intentionality around what you're going to get out of it. And this leads to, a strong digital backbone development and deployment leads to interconnection across multiple digital technologies and, those more rapid and deeper discoveries. So what are critical elements to R&D transformation? Well, we think there are sort of 3 pillars at play here. And it all revolves around information at the end of the day. The first is, they're not really in any particular order, but first is process visibility that, as Tim was highlighting from our previous days at Amyris, if you don't know how something was made, you don't really know much about it other than its state in time. And that's not very helpful because it means it's incredibly difficult to reproduce that production, that thing again. Another component is data usability. And the panelists were chatting a bit before we let everybody in. And, this is sort of near and dear to everyone's heart, that more accessibility and more usability of data is key to us transforming the way R&D and digital capture is done. And finally, quality, which is a big word that means a lot of different things to a lot of people. But I think at the end of the day, you can summarize all of this to mean access to the data and process information, and trust in that data are core to your success. That's really what sits central to all of this. So why is the digital backbone so valuable? I hope this kind of answers itself after what we've just talked about, but I'll spell it out in any case. First, it's going to enable meaningful context-rich data capture and integration. That holistic 80% capture or 80% data problem goes away because it's endogenous to how you capture data. And that holistic data capture is critical to determining cause and effect relationships. You know, as we all know, the modern lab is highly multidisciplinary, and it means that relationships between effects can be much farther reaching. Things that are happening in molecular biology are playing out in you know, product finishing, and without building a data model that connects those two, there's really no way to discover that. And complete data capture is really the only way to fully articulate a system. It's the only way to know everything you possibly can. It provides a scaffold for all participants in that research to work from, and hence a shared view of the activities. And finally, and you know, most importantly for the business, is it's going to accelerate work, reduce redundant effort, and lower the cost of that effort. So I'm going to dive into a case study from a previous company. I won't name names, but I'm sure you can figure out who I'm talking about. So at a previous company I worked at, there was a significant effort to make a structural protein and convert it into downstream materials. And this was a highly, highly complex scientific workflow, starting from molecular biology and protein engineering through fermentation and purification. It included a scale-up and tech transfer component to make sufficient material to actually do mid-level scale testing. Material processing was in the mix, and of course, final materials characterization across every one of those verticals. And the problem was we had no comprehensive data solution in place. We were working from Excel files, Google Sheets. You know, you were lucky if people actually captured data on each unit op, and there was certainly no interconnectivity. And the work to get that data together was, you know, as we all know, incredibly difficult and very time consuming. And so we undertook transformation the hard way. We produced a holistic process and data model in Google Sheets, which is, you know, a little archaic, but at least it's there. And this was used to capture key process information and data from each of those unit operations. We included Python-based form entry for capture of material prep details. And so this was to simplify scientists' entry of data so we wouldn't lose as much as we had been in the past. And I'll point out that even with this, it still required a fair amount of manual curation. So, you know, the punchline is that you still, even with something like this, there's still so much more opportunity to make it better. But even with just something as simple as an organizing principle like this, we saw a 20x improvement in product yield, a 4x reduction in impurities, a 2x improvement in material properties of the final product, and, you know, I say infinite, but an unmeasurable increase in efficiency and capability. And it was that discipline and consistency in data and process capture that allowed us to uncover the hidden trends and drive new hypotheses. A lot of the things I'm going to show you on the next slide in terms of interdependencies may not have been studied if we hadn't been assembling data into this newfound analytical scaffold. So it's really that the data capture mechanism drove the types of scientific questions that we were asking, because we suddenly had more line of sight on what was actually happening. And so we could look at a lot of those interdependencies just as a smattering of examples here. You know, between molecular and biology, you discover that, I mean, I think this is a no-brainer for a lot of us, but gene copies were heavily impacting yield, but now we could more explicitly understand exactly how. We had a protease problem and we needed to inhibit them, and that helped improve the product profile coming out of fermentation. Between fermentation and downstream processing, learning that there were a lot of process conditions that had to be tackled. In this case, we needed to neutralize our final broth against degradation and manage hold times. This enabled, between fermentation, DSP, and scale up, a derivation of critical process parameters, KPIs, and so on, understanding processing time, and understanding robustness. And finally, from end to end, we were able to derive purity and quality impacts on each stage of production. And that helped us build out a master recipe capability of tolerances and limits on each one of these steps. That was really key to ultimately improving the product to a level that it became sellable. And so for those of you who know which company I'm talking about, you know what kind of products came out of that. And, you know, these were a direct result of this sort of interconnected digital scaffold work that we put into it. So as great as this was, I came to a point where I realized that there was still a lot more to be done in this space. And fortunately, Tim had reached out to me and there was a role available at Riffin. And I thought, well, it's fun to do this for one company, but it could be even more fun to develop a technology that would support any company trying to do this. And so I jumped over to Riffin. And in my talk, I'm going to jump over to Riffin and talk about what our goals are and what our the 3 pillars of Riffin Nexus are. So as John introduced earlier, Riffin Nexus is a process data system. And it's all about placing the R&D process at the center of everything. And it's aimed at solving data usability and system flexibility problems that live in your traditional ELN, LIMS, and scientific data management technologies. And the punchline is that you get integrated analysis ready data instantly, and we aim to be the hub of your R&D data ecosystem. So pillar 1 is that we are process centric, which means flexible but structured processes are providing meaning to your scientific data. Again, this goes back to that refrigerator conversation or anecdote where if you don't know how you made something, you don't really know much about it. And so if you look at the space today, you've got LIMS, which tend to be sample centric, and that's exactly what Tim and I were looking up bottles out of that refrigerator in, was a LIMS platform. And you had a hit or miss success rate on actually learning anything about those materials. On the other end of the spectrum is the incredible flexible electronic lab notebook, which tends to focus on individual experiments. But there's a place sort of marrying these 2 concepts where you take both the structure and the flexibility to define your process. And that's where Riffin lives. And for those of you that are curious to know more about exactly how we do that, I encourage you to stick around at 9:30 and join Andrew Dalton and Josie Livingston for a demo. Pillar 2 is the ability to pull data together for analysis in real time. And so the idea, all of those different pillars and technical verticals that I referred to earlier are generating data of different types, heterogeneous data. And so Riffin Nexus enables the collation of all of that heterogeneous data from a variety of sources. It can come from instrument software files, from external databases, LIMS, and historians. You can push data in or pull it out through the API. And of course, you can enter data manually. And the magic is that it's taking that data and putting it into a statistical data frame and porting it over to any analysis platform that you're interested in. And that could be done directly through the UI, downloading the data. It could be done through the API, and it could be done through a SQL query. And that information can be, pushed directly to a platform like Jump, like Spotfire, or perhaps to your corporate data lake, and then from there into Python notebooks and so on and so forth. And so really the magic here is at the core, where it takes all of this different data, uses those process maps to create a contextualized data frame. And that's really key. And this is constantly updated in real time and available for download at a click. The third and final pillar is that we aim to be the organizing hub of a modular data system. We understand that you need a lot of different technologies to accomplish today's R&D requirements, and no one solution is going to solve that. And so our goal is to be open and play well with others, and that's exactly what we do. through a variety of means. And so Riffin is meant to be the workflow data capture and structure capability that can play well with external ontology systems, master data and molecular design systems, connect into your primary instrument data capture systems, and then push data to data lakes and your analytics platforms as needed. So I will leave you with a final thought that I was mulling over this morning, and maybe we can take this into some of the discussion later. I think it's ultimately the case that how you collect and store process information and data drives what gets captured. And so your digital backbone creates possibility. And the goal is to engineer as much possibility as you can. Thank you.
Fantastic. Thank you, Lauren. We have a question that's come in from Chung Kai Lim. Chung Kai says, how best do you think we as research scientists in the lab with experiments that fluctuate and change on a day-to-day basis can best integrate platforms such as Riffin into our workflows?
Well, first, I would invite you to sign up for our open access capability, and that can be done on our website. I think what you'll see, one thing Riffin does really well is it's a highly modular platform. So it allows you to deconstruct a series of scientific steps into individual sub-processes and then string them together. And so if you think about it, while your day-to-day may change, a lot of the tools that you use are similar. It's just the order that you put those together. And so, you may run one assay first and then use those results to trigger a second assay, or you may run a particular, molecular biology methodology first, and then do a purification downstream. And so you can string these different modular components together in any order that you want. And that's something that Riffin does particularly well.
Great, fantastic. So we're going to come back to you in the discussion, Lauren. But next I want to welcome Elizabeth Irons. Elizabeth is the CEO and the founder of Science Exchange. And Science Exchange is an online platform for finding any kind of scientific service that you might wish for. So Elizabeth, welcome to SynBioBeta Live. Elizabeth, can you hear me? Are you still on mute? Well, we'll come back to Elizabeth in a second. Looks like we might have some connectivity issues. We also have a poll that we wanted to ask our attendees. So if we can start that poll, we'll be able to gather that data and share it with you. But next we have Ken Finley. Ken is a generalist in industrial biotechnology. He was trained in molecular and cell biology and developed a passion for bioinformatics, working on interesting yeasts. Ken is a leader of data strategy for Cargill's Biotechnology R&D Center. Ken, welcome to SynBioBeta Live. You're just on mute, Ken. So if you want to unmute yourself, we can dive in. How are you?
Yeah, I'm great. Thanks for having me. Super happy to be here.
Great. And I'm sure everybody's heard of Cargill. I believe it's the largest private company in the US. I wondered if you could just give us a sense of the breadth of Cargill's business. What are the big chunks that Cargill does?
Well, Cargill got its origin started in the corn milling days out of a cooperative grain exchange in small town in rural Iowa. Grew from there to be a big player in commodity spaces. Food and feed ingredients are primary products that Cargill puts out, a lot of high intensity sweeteners. Cargill's moved beyond its food roots to some extent and has big plays in trading now and a lot of sustainability efforts across a variety of commodities in the world.
Great. Can you tell us a little bit more about the sweeteners? What are the sweeteners that you mentioned?
So one that you might see on the market is called Truvia. That's the Cargill branded high intensity sweetener that occupies pretty large niche in the high intensity sweetener space, and you'll find it in a large variety of your favorite beverages, on low calorie favorite beverages on your grocery shelves.
Great, excellent. And which products specifically are you involved in, or are you more on the scale up side?
Neither. I'm on the R&D, the genetic microbial engineering side, also metagenomics. So within biotechnology R&D, we support animal nutrition businesses that are and human health endeavors that are now branching out into metagenomics and what kind of gut health outcomes we can achieve.
Great. Yeah, I've taken the COVID pause as a opportunity to look at my diet, and I've been experimenting in all sorts of different ways and things that I eat, and I have been overwhelmed by the impact that that what I eat has on my mental health and on my gut. The strength, the connection between those two things is I always knew it existed, but I had no idea it would be so powerful. Things around gluten and lactose and sugar. So really quite remarkable, the connection there.
Indeed.
So the survey that we just asked everybody, what is your biggest challenge? And the results are 54% of people said data cleanup and organization. Can you talk a little bit about that? And I think you're using Riffin Nexus, and maybe talk about some of the things that happened in your organization after you started implementing it.
Yeah, certainly. Data, must be a lot of data scientists on the call, I would guess, from that response. You know, it's we're transitioning from largely Excel driven. We have, you know, tremendous volumes of data scattered across Excel spreadsheets, and they're just not formulated in a way that lends themselves to long-term historical analyses. So we're using Riffin now to start building those structured data sets where the data will be there for a long time. So we're looking at the granularity that's available to capture in the system. We get, as Lauren just mentioned, we can capture enormous amounts of detail and set the stage for more longitudinal, deeper dives into the data that should enable more robust discovery and really expedite our development cycles.
Lauren talked about the FAIR guiding principles. Can you talk a little bit about FAIR? What does it stand for, and what does this mean in the context of data?
Yeah, sure. FAIR is data should be, it's a principle and a philosophy that data should be findable, accessible, interoperable, and reusable. And so that means that I, as a scientist and somebody who comes after me, should be able to access the data quickly, load it into whatever algorithm, machine learning model, access it very quickly, and use that data without a whole lot of barriers. And it needs to be, you know, highly available in a pretty, you know, basic format. Quite frankly, tidy data concept, rows and columns, is today what works best.
Great. And what one big idea in your field do you wish that every non-scientist understood, and why?
Would be that genetically modified organisms are going to be required to feed the human population in the future. You know, this is one of the places where maybe it's a U.S. science policy problem that science is falling a bit short, and I think we can see some of the ramifications. Population being as big as it is, genetically modified approaches extend well beyond human health. You know, I get the alarmed questions from friends and family. Oh, my lands, did you see what is going on in human? Well, we don't play in that. Inside Cargill, my world is consumed with what's happening in microbial spaces, and I'd say that there's probably a lot more going on in the microbial space. Well, that's probably not a fair statement, but it feels like to me, certainly there's a lot more going on in microbial spaces than in human health segments.
And how has the pandemic impacted Cargill?
Well, locally for us in the lab, it started out with a majority of folks working from home. Projects were prioritized given the number of food ingredients that Cargill supplies in the world. Critical to support the production manufacturing facilities and keep that R&D work going. Of course, that all happened with social distancing and increased PPE. We look like a BSL-3 lab now, and even though we're, you know, more BSL-2 lab, and although we don't do a lot of pathogen work specifically, it's more for unknowns. And then it's slowly been opening back up. We've kind of reconfigured the lab as to what are available workstations. Of course, Cargill's product lines, protein was a good example in the meat spaces where the meat industry was walloped badly by the the close proximity of production workers, and lots of plants were coming offline. And of course, that was a huge critical problem for Cargill. But at the same time, the alternative protein space has gotten a lot of traction, probably seeing a lot more growth in the alternative meats like Memphis meats than would have happened otherwise without COVID. So certainly the jury is still out on the total financial impact right now, because it continues to play on, but I think we're recovering nicely, and we'll see.
Great, excellent. Well, thank you, Ken. And we'll come back, Ken, into the discussion and talk a little bit more of the technical details and how you're actually using Riffin and some of the benefits of it. I want to bring in Elizabeth Irons. Sorry, Elizabeth, we lost you there at the critical point where we transferred over to you. But as I said, Elizabeth is the CEO and the co-founder of Science Exchange, and she's the co-director of the Reproducibility Initiative. What is Science Exchange, and how has it benefited science and technology research?
Thanks, John. Apologies for my technical issues.
No worries.
So Science Exchange is a platform for outsourced research and development. So what we provide is a marketplace that allows scientists to work with any CROs, CMO, or core facility without having to set up a contract or qualify that facility. So really trying to remove all of the administrative burdens associated with finding an external partner to conduct research for you, setting up the ability to work with them, so no contracts are needed, and then being able to manage and pay for that project through the platform.
Great, excellent. And what is the Reproducibility Initiative?
The Reproducibility Initiative actually came from Science Exchange's mission, which is to improve the quality and efficiency of scientific research. And what we wanted to do was use the network to find a way to be able to easily replicate scientific results, and by doing so actually provide a way to assess the reproducibility of published results. And so the Reproducibility Initiative aims to look for ways to assess the reproducibility of published results.
And can you give me some of the results from the Reproducibility Initiative, and
Sure.
what's going on, and what's the plan for the future?
Yeah, so the Reproducibility Initiative has been involved with most of the major reproducibility studies that have been conducted in the biomedical science space. So we ran the Reproducibility Project Cancer Biology, which is probably, I think, the best known reproducibility project. And that was an attempt to replicate the key experimental results from 50 published cancer biology studies. That project is actually just completing right now, so we're in the final publications from that. And we've also there's several other reproducibility projects that were run on a smaller scale for the Prostate Cancer Foundation, as well as an antibody, an independent validation of antibodies. So to look at whether antibodies as a reagent could be independently validated as a quality reagent.
Oh, great. Are you doing anything related to COVID antibodies?
No, that project has been completed. I've definitely seen a lot of the data coming out about the quality of reagents related to COVID, which is an area that I think is quite problematic at the moment, just like most antibodies. So yeah, it would be important to make sure that those are properly validated before use.
Great. There's somebody, I can't remember which university they're at, but they're called Erica. We had her on the line the other day. She's doing a comparative study of all the COVID antibodies for the Gates Foundation. Do you know who I'm talking about?
I don't know, but I saw a study that was run, I think, out of the Chan Zuckerberg Initiative that also compared all of the serological assays that were being used and looking at their specificity and sensitivity.
Great. So give us an example of how big this problem is. What did you learn from the cancer study or the prostate study? What is the problem?
Yeah, I think reproducibility is, an area that's probably, very much a topic that everyone on the panel is familiar with, and a lot of our work is designed to, make it easier to actually get high quality reproducible results. I think publications as a tool to use as a way to communicate scientific work that can then in theory be built upon and reproduced is very problematic. So we found that most published studies, you can't really reproduce the results. And that's not really that the results themselves are not reproducible. It's more that the publication doesn't provide sufficient information. There usually isn't, a lot of the key protocols, reagents, etc., available to really be able to take those publications and reproduce those findings.
Tim talked at the beginning about human structures and organizations. If you could have your druthers and tear a few things out, what would you look at in terms of reformatting our human structures and organizations around scientific reproducibility? Where would you start?
Yeah, I think there's a lot that we could do, and we were talking about this before, the call started today, but I think it's really today we still a lot of the time exist in a relatively artisanal science world, but we're making that transition to a much more industrial science world, and that's actually really exciting to me because for me, I think that that's really what will drive the improvements in reproducibility. When you look at the scientific methods that are actually highly robust and reproducible, a lot of them are the industrial methods like next-gen sequencing, areas where there isn't so much human hands that actually then have all of these uncontrolled variables associated with them. So when I think about tearing things up, if you were to try to create, as much benefit from the scientific data that's being produced, you probably would try to use machines as much as possible to produce the data. You would try to have a data store that was available for everybody to be able to analyze all of those results and be able to use all of that background information as soon as possible, rather than, this idea of, storing it all up and then having this nice, small amount of data that's put together for a publication.
And if people aren't familiar with Science Exchange, there's the ability to go on the website. You can type in any kind of service that you want, from DNA sequencing. I know that was one of your early things that you focused on, to any kind of test or assay or use of a machine or core facility. Where are you seeing the most growth? Which kinds of, where are the biggest chunks of things that people come to your platform to outsource, and why? And then what are some of the trends that you're seeing on the platform recently?
Yeah, it's really interesting actually. So Science Exchange, a lot of people know about our reproducibility projects, which is, probably why you talked about them, but actually, really the vast majority of usage of Science Exchange is as a platform in pharmaceutical and biotech companies to provide a way for them to centralize and remove the administrative burden associated with outsourcing for their scientific teams. So up to a third of scientists' time is just spent on administration. So how do they know if there's a contract in place with who they want to work with? How do they know if that external team is going to be good at doing the experiment that they need? How do they track where they're at, in terms of delivery, and how do they figure out whether their organization is actually paid for each of the milestones that have been completed? And so all of those steps really take away from the productivity of the scientific teams. And so, the vast majority of what scientists use, Science Exchange for is really just the run-of-the-mill management of all of their outsourced research and development. And for me, what's been really interesting is that I always hypothesized when I started the company that there would be more and more dependence on a networked approach to R&D, where people were really outsourcing to specialized facilities where they could use things flexibly on demand. And that's really come to be. So most companies now have more than half of their R&D expenditure actually conducted at contract research organizations and manufacturers, rather than on in-house R&D teams. And so in terms of the types of experiments we see a huge range. So it could be anything from antibody engineering through to clinical development, central lab services, through to real-world evidence collection. So there's very, very wide range, I think now over 7,000 types of services that are offered through the platform.
Fantastic. Well, I'm certainly glad that you exist. If I was back in the lab, I'd certainly be designing all my protocols around around outsourcing. I'm curious how the pandemic has affected your business. What are you seeing, the last 3 months, and then what are your predictions for the future?
Yeah, we actually have an R&D advisory group, which is made up of heads of R&D of biotech and pharmaceutical companies. And so we've actually met regularly through the pandemic, just to have an opportunity for those leaders to talk to each other about how they're dealing with the pandemic. And for us, what's been really interesting is that, most R&D organizations actually did shut their research to some extent. So they either prioritized certain programs that were either related to COVID or critical to their organization, but they really focused on, maintaining manufacturing in particular, while actually shuttering some of their research programs. And what was interesting was they then moved a lot of their research to CROs. So CROs remained completely operational. So Science Exchange, uses our data to look at business continuity within all of the 3,000 CROs and CMOs that are operational through Science Exchange. And over 95% remained fully operational through the pandemic. And so really what we saw was, however, that there is issues with actual supply chain. So there's issues with the CROs and with in-house research organizations being able to obtain critical reagents, mostly reagents that were produced in China. And that actually has caused, significant delays in research projects being able to be completed. So it's been interesting to just see that reliance. And I'd say one big change that I think everybody is sort of aligned around is that there won't be any sales reps or any of that type of in-person selling process allowed in biotech and pharma organizations in the indefinite future. And so for us, one of the things that we really focus on is how do you provide an e-commerce or an online interaction with all of these external facilities that you're working with. And so that's been a critical path is really how do people move from this offline sales process to an online process where they're working with their CRO through a platform like Science Exchange.
Fantastic, that's great. And there's all these hidden things that you don't think about reps going around to labs and that not existing, at least in the foreseeable future. Great, thanks for that insight there, Elizabeth. We're going to come back and have Elizabeth and Ken join for the discussion shortly, but I want to bring in Jessica Louie, and Jessica is at Join Bio. And Jessica, if you don't mind unmuting yourself and turning on your video. And then also you've prepared a few slides, and I think that would be nice just to get a quick overview of Join for people who don't know who you are and what you do. So if you don't mind sharing your screen, then we can dive in.
Absolutely. Can you hear me okay, John?
Everything sounds good. And I will remind our audience that this is a live event. And if you want to type in any questions to ask any of the panelists or join in the discussion, please type them into the Q&A box. I see we have a couple of good questions coming up, which I'm going to save for the discussion. And I'd like to bring you in person. So if you raise your hand, we'll bring you in and you can ask the panelists your question live. So Jessica, over to you.
Great. Are you seeing my screen with my slides?
Yeah, we got it.
All right. These days with multiple monitors, you never know what you're going to share.
It's all good.
Great. So just a few slides. And as John introduced, I'm the data science lead at Join Bio. Join Bio, we launched in 2017. Our parent companies are Ginkgo Bioworks and Bayer Crop Sciences. So with our two parent companies, we leverage Ginkgo's Foundry services to engineer microbes, and we leverage the Bayer Crop Sciences ability to test those strains, our engineered microbes, for agriculture on plants, and also the agricultural know-how. So we exclusively license Ginkgo Bioworks technology for agriculture, and you may have heard that our initial programs are focused around nitrogen fixation, and our long-term goals are around building a platform for agricultural innovation. Other directions that we're thinking to go in are around crop nutrition, pest protection, and disease management. Around our platform, we have the concept of chassis payload leading to an optimized genetically modified microbe. So a chassis is a microbe that exists in nature and whose qualities make it viable to genetically engineer, and also as a viable for our product concepts because they are relevant to agriculture and they associate with plants or crops of interest. And then payloads would be the gene cassette or the operon, which contains proteins, which will express proteins that can confer some efficacy against our product concept. In the case of nitrogen fixation, it can be nitrogenase activity, and in the case of, say, pest control or disease management, they would be relevant to, those would be payloads relevant to killing insects or other sorts of microbial pests. And then we take those and we test those. We take those and we test those optimized microbes on plants, ideally if we can seed treat, and then assess colonization and efficacy against our product concept. So finally, that's where we're headed at Join Bio in terms of building our platform. And here's a snapshot of our team. We are a small company and a startup, and we are we're very happy to be working across Boston and across California.
Excellent. Thank you, Jessica. And where are you based specifically? Are you in California or are you in Massachusetts?
I am in Massachusetts.
Great. Excellent. And are you a Riffin Nexus user?
We have done a POC with Riffin, and that was actually in our California site, where prior to the pandemic, I was very frequently visiting.
And can you tell us a little bit about your work as a data scientist? What do you do? What kind of data are you dealing with? And how did the proof of concept go?
Yes, absolutely. My work is a data scientist, so I lead a team of both biostatisticians and data scientists, or really hybrids. And our goals within the organization are to ensure or shepherd through our work streams the veracity of data, speaking to sort of the 5 B's of big data. When thinking about R&D data, I identified that veracity was really the challenge as opposed to volume. We also have variety, but volume and velocity are not as big of problems as they would be, say, in high tech. But veracity, and that's where our biostatisticians come in to play a key role in helping to standardize our processes, reproducibility, centralize on objectives and experimental questions, and ensure that we have an understanding of our confidence in experimental data that are captured. In terms of our interaction in our POC with Riffin, I think that through our process of interacting with Riffin and evaluating Riffin, it drove us to really centralize and standardize what we're trying to do with our experimental workflow. It was more ad hoc prior to our engagement with Riffin, and it made us go through a process of introspection and partnership together with our R&D scientists to mature those workflows and understand what we're doing and what we expect to get out of those assets.
Great, excellent. When you're seeing new people joining the Riffin team, maybe they're coming from academia and you're trying to train them in the way that you think, where do you think the biggest skills gap is, or the different conceptual gap between what they've been doing in academia and then what you're trying to train them to do now in industry, particularly around data management?
Yeah, that's a really great question and really relevant, because within our Riffin POC, our lead scientist was in fact directly from academia, where he had postdoced at UC Davis and then previously from ETH. And I think in that case study very specifically, taking a structured approach, both to sort of an assay development process or like experimental research, even exploration of how to develop that assay or how to run that assay, taking a structured approach and a phased approach, and then evaluating assay quality they were somewhat foreign concepts. And also sitting down and planning objectives, factors to test up front prior to getting in the lab and getting hands-on. Focus on the quality and veracity of data as opposed to the key results that might be presented in a meeting. Like just putting a graph on a slide was really the focus prior, and also for positive results. I think there's a large drive in academia to receive a positive result. to support a positive conclusion, that is, as opposed to, say, a negative conclusion. And in thinking, zooming out from assays, but when we think about what we want to learn about our system from our DBT cycles, we care just as much about negative information as we do about positive information. And in terms of machine learning and AI, those negative labeled examples are just as important for training our algorithms. And so I think that was a key gap. In terms of data management, I think working out of Excel sheets versus templates, or which can be represented in Excel as well, but structured such that a machine can read them, keeping standardized data formats. These are really low level aspects, but certainly the very first foundations and first steps.
Great, excellent. Well, I'd like to bring in the rest of our panelists to have a discussion around this. So if people want to turn on their videos back again. And Elizabeth, I just wanted to come to you to talk about what we just talked about in terms of the incentives that people have in academia versus the incentives that they have in industry, which Jessica was really saying, well, you're so much pressure to publish something in academia to kind of get the handcuffs off and get out. But then you jump over to industry and there's more pressure to be right than just to get an answer. Do you agree with that?
Yeah, I definitely agree with that. I think the incentive system in academia is extremely flawed, and I do think it's very motivated around publications, particularly in high impact journals, and exactly to Jessica's point, this kind of positive publication pressure of really just publishing positive results. And I think also there's a trend towards very, very long kind of stories of data that have 50 different experiments. And I often wonder how many experiments they did in order to get that sort of nice 50 experiment panel, and there's probably 1,000 other experiments that didn't kind of quite match the story that aren't in there. So the sort of presentation of results, particularly from academia, I think is very problematic.
Great.
So I'm curious what you would do differently, Elizabeth. What would you incentivize, and like how can we graduate really strong scientists out of graduate programs? What does that look like in the future for you?
Yeah, and even that point around the difference between industry and academia, for a long time, industry actually didn't want to hire PhDs necessarily out of academia. They would rather hire like masters trained because they felt that they would have less kind of bad habits they had to undo, which I think is kind of shocking. So I think there's a really strong need to train scientists on statistics, so experimental design, digital strategies. So one of the things that I also kind of find shocking, particularly as the CEO of Science Exchange, is this really strong concept that graduate students should do everything themselves with their own hands, and they shouldn't outsource any of their experiments, and they shouldn't use kits or anything that are actually validated reagents. So it's that kind of bothers me a lot, because that's the reality of the world they're going to go into as scientists. They're going to have to be able to manage outsourced providers of data that they then have to analyze and incorporate with their own data sets. They're going to have to be as fast as they possibly can and have high quality results. And so yeah, if we were to change things, I think it's moving away from this emphasis on publications as the only unit of output. So is there a way to start to really capture much more of the products that are produced in the lab, either through more automation and machines that generate that data, or through artisanal experiments, but so long as they are captured in a data store, and then have measures of output, quality of design, reproducibility of those results, and the whole package that's generated doesn't necessarily have to be some very unexpected breakthrough. It can actually just be a body of work that tested an interesting experimental hypothesis well.
So you wouldn't.
Yeah, go ahead, Lauren.
Well, so I guess the punchline is, even if the result was the answer is no to my hypothesis, as long as I defended it well, it's still a good outcome.
Exactly.
I just wanted to bring in, we have a guest joining us, Anton. Maybe you can unmute yourself and tell us where you're coming from. And then you posted a really interesting question where you described your current workflow, which I think many people will will resonate with. And then the question was, well, what would it look like in Riffin? So I wondered if you could just introduce yourself and give your current workflow.
Sure. Hi. Thanks a lot, John, for bringing me in. I'm currently doing PhD at the University of Edinburgh in the UK, and I certainly can relate to all those things that you guys mentioned. And I unfortunately have to live through this myself, with all those problems and data management and the way experiments work and the way we have to think in this kind of a box, which is very, very different compared to how industry works. And to be honest, like from my perspective as a PhD student, looking at the things that are being happening in the industry, I just feel the academia is just gonna die because the space is just so far. I mean, what you can do with this stuff, you know? It's not even like even before the data, the way to generate this data, it's all still manual. And once you have this data, yeah, you have to do with all these platforms, all the systems, and it's all manual. Because at the moment, if there are any alternatives, I'd be happy to learn about them. And Riffin sounds absolutely amazing to me.
So just talk through, Anton, that you talk through the 6 steps just very briefly that you posted into the chat. You're starting off by collecting data. Yeah, go ahead.
Hold on, I'm trying to find the Q&A.
And you're collecting what kind of data? Is it sequencing data?
Yeah, so at the moment, so basically the particular experiment that I was talking about is fluorescence data. So imagine I have some sort of a dye that I'm testing at the moment on and trying to figure out the thermal stability of a dye. And I'm using fluorescence for this. So I have like 4 different biological replicates and I'm doing measurements. And this generates a bunch of data in the format that is being given by this machine that does all the analysis. And I love this because otherwise I'd have to do it manually. At least I can do multiple reps at a time. So I'm very grateful for that. But the format, of course, that the data comes out is not compatible with the rest of the downstream data processing. So what I have to do next is to come up, well, first of all, figure out how does the software works that I'm using to process this data, and then try to basically recompile, you know, this like hundreds and hundreds of columns of data in this Excel or CSV file into something that would allow me to break down my data per sample or per biological replicate or technical replicate, and just try to basically put it together in another kind of spreadsheet that will then I import into this time. So all of this would be done in macOS, which I'm usually using for analysis. But then I have to move it to Windows because some software is not compatible across platforms. And I would use Origin Pro, which is a much more sophisticated statistical software to analyze all this data. And after that, you know, I can actually visualize all this data for the first time properly, you know, graph all of my errors and so on. And then after I see that my data is compatible and the results are significant, for example, I would then resave it again, this time in macOS, and then try to use different software because plotting things in Excel is just so painful. I would then use, it's called GraphPad, to actually present the data in a way that would be actually publishable and easy to manipulate the graphs.
Great, thank you, Anton. I want to bring you in, Ken. Is this just something that's in the corridors of academia, or do you think there's people in industry who are struggling with the same issues that Anton described?
There's absolutely people in industry struggling with the same issues. Where Anton's off to a really good start is having laid out the workflow. And that's the kind of thing that really lends itself well to Riffin, because he can take the various steps, configure the workflow, capture all the relevant data, end up with that single unified data model, and then figure out the visualization. I think for the most part, Python there, and maybe a little bit of R to help with the statistics, or JMP if he prefers a Windows platform. You could get a ton done a whole lot faster. Just, you know, go to the end of the problem and say, what's the question I'm trying to answer, and what data do I need to support it? How do I need, what information do I need in rows and columns? And then all of those modern software applications will not care that much about the order of things, and you select your variables and you set up your algorithms and you're off to the races.
I'm curious, Anton, what part of your workflow do you spend the most time on, would you say?
It's a good question. It really depends, because as a PhD student, this might be the experiment that I'm doing for the first time. So there are lots of things for me to figure out. But I would say in this particular example, it took me a while to get back to Python and actually compile a script, because this is something that I would not do that often. And obviously, like if I do it maybe once or twice a year, I need to kind of refresh my brain how it works, and then different standards, etc. So that takes a while, yeah.
So that's the thing I think Riffin is really well suited for. And I'll actually let Ken, sorry, I'm interrupting you. So Ken can speak to that in droves, because that's what we're doing with them.
Well, my comment was going to be, you're teaching yourself to fish on the Python front. So the longer you can stick with that, the better off it's going to serve you. Python seems to be king right now. I imagine that to persist for a while. And, then the other place that you do yourself major favors is feeding the structured data. Just get it into rows and columns, and then you don't have to touch it as much, and you get to spend a lot more time on the fun science questions evaluating the data.
Yeah, I absolutely see how my approach could be optimized. And, even with Python, I can do all the charts and plot everything, or use R for the same purpose, and just kind of remove all the steps downstream and get from the data to the graphs, like almost immediately. But then, I mean, that would be another investment of time that I need to do. And since this is not something, this is just one of the, maybe 8 or 10 experiments that I have to do, I need to justify how much this investment of time is, meaningful in this particular context. And my supervisor might not necessarily, kind of support all the struggle, because he or she might not even care about all of this, They just want to see the results. They just want to see the graphs. And all this hustle in between is just kind of, it's my personal problem.
Right. And if panelists, if you just want to raise your hand, and I'll try to moderate and make sure everybody can come in. Yeah, Jessica, go ahead.
Apologies for that.
No, it's all good.
Coming maybe somewhat proximally from a graduate experience, I went into a career in data science, but as an experimentalist, and also my PI likewise would not have cared about anything that happened in between, and also would have only wanted to see a positive result, in fact, not any result. I can definitely understand where you're coming from around learning Python and the friction that can cause, or just the amount of effort that can cause. And having done recently a Riffin POC here at Join Bio, I wonder if there is space for Riffin, even in a more flexible setting. I found it fairly straightforward to lay out workflows in the workflow designer, even if it wasn't, even if it was something that I would only really consider doing one time. It seemed straightforward enough to set up the process, and I found it could be a really great way to merge the data from the different steps, and then the output would look like a tidy data frame or Excel output that could be exported. And I think Lauren spoke about analyzing that in JMP or in Python or R, but actually having used GraphPad quite a bit in my actually back in MIT days. So I think that it could be opened in GraphPad very easily as well.
We produce vendor neutral CSV, so you could open it in anything. And I would add one comment, just not to belabor the point, but there's probably value in laying out your experiment. You said, Anton, that you're often doing something for the first time. And so building the Riffin process could also be an excellent way for you to sort of discover what it is you're trying to do, because you have to think through what am I trying to answer, and what information will I need to answer that. And so it'll drive you to build a workflow that builds the right experiment.
Yeah, Lauren, that was exactly our experience in our POC with you. Is building our workflow really made us hone in on what is it we're exactly trying to do. And we were able to iterate on each workflow. It's a great point.
Excellent. Anton, thanks for joining us. Lauren, if there are students out there who want to start using Riffin in for their PhDs, and then hopefully they'll become future customers of Riffin when they move into industry or when they start an academic lab, how do they do that? What are the entryways? And then also if there are startups out there or companies who are saying, that's me, I recognize my problem in what Anton's saying, what's the best way for them to kind of start and get a handle on the platform?
So we actually offer Riffin for free to academic programs.
Oh, great.
To academics. And so if you go to riffin.com/demo, you could sign up for our open access. It's towards the bottom of the page, and we'll get you an account. And there's a whole bevy of free help content once you're a registered user, which would include a video of our super user training. And so you could walk through all of that and have a pretty good handle on how to use the system.
Excellent. Great. Thank you, Lauren. And I think we have another poll that's going to come up right now. GDPR rules mean that we can't pass your information on to Riffin unless you give us permission to do that. And so the poll will come up. And if you want to allow us to pass on your information to Riffin, Lauren, I think you've agreed that you won't spam people. Is that right?
It's right. We're judicious in our use of email.
Yeah. So, but if you do want to contact Riffin and have them reach out and offer you a demo, and how you can set up. Also, in about 15 minutes time, there will be a demo where you can actually go deep on the technical platform, and there'll be another breakout room that pops up in the chat for you to jump to in 15 minutes time. We have another caller dialing in, Ravi Ram from India. If you want to introduce yourself, Ravi, and then you think it's an attitudinal problem between industry and academia, is that right?
Yeah. Hi, John. How are you doing?
I'm good. Welcome.
Fine. Yeah,
Thanks for joining us.
Ravi from India, South India. My city is called Chennai. So I lived both in US and also in Europe in the academic side, and moving back to India, worked more on the industry side. Okay, from diagnostic development, also agricultural, biotech. So my question is, see, reproducibility, I think is, as I mentioned in the question, it's an attitudinal issue. There's a lot of difference between academia as compared to the industry. So you want to adopt all these practices in industry, it's more amenable. How do we promote this in academia? Now that's a larger question, I think. And does the platform, of course, kind of help channelize the adoption of these practices in academia at the level of academia itself? And also, philosophically or phenomenologically speaking, do you think that these needs to be adopted more streamlined in academia, or there's more room for freedom?
Great. Elizabeth, do you want to take that one?
Yeah, I definitely 100% agree with you, Ravi. I think there is just very strong cultural differences, but I think that the culture comes from what's rewarded. So by definition, you kind of get what you reward. And so in academia, I think there's a really strong problem that really what is rewarded is high impact publications, but those publications have no measurement of quality whatsoever. And the few studies that have been conducted on the reproducibility of published results, or even the statistical design of experiments in high impact journals, indicate that a large number of them are statistically flawed, that most of them are not reproducible, and that there's no correlation with the impact factor of the journal, or even the number of citations that the individual paper has, and whether that result when measured independently can actually be reproduced. So I think if we want to have a better quality of results that are produced from academia, we have to actually use a way to measure the quality of those results. And so far the funders are very much unwilling to really tackle that on head on and actually be able to measure this. So the only one that I'm aware of is DARPA, which just recently published that they are funding from the biological division the independent verification and validation teams, which will actually work alongside all of their funded researchers to independently validate the results that are generated in real time. And that's a much better method, in my opinion, than trying to take a published result, which is usually years after the work was actually done, and be able to go back and try to reproduce that when usually the postdoc or PhD student who did the work is now no longer in the lab.
Great. Thank you, Elizabeth. And thank you, Ravi, as well. We have a question from Mohammed Eslami. Mohammed, do you want to turn on your video and unmute, and you can ask your question live? Yeah, go ahead if you want to introduce yourself, Mohammed.
Yeah, hi, my name is Mohammed Eslami. DARPA was just mentioned. We're actually a performer on one of DARPA's synthetic biology programs called SD2. And so we face a lot of these issues in, building these kinds of workflows and being able to analyze data across multiple labs in some kind of unified interface quite a bit. And so the question I have, though, is we find it very difficult on the program to be able to objectively measure if we are indeed increasing productivity, right? And some metrics that we've tried to use are number of analyses that have been run over a current week, the number of data sets that have been processed, to really see if the automation that we've kind of baked in, the automation organization that we've baked in, is actually increasing our productivity. But we don't have good baselines for those to really come and say, are we indeed getting better? Are we moving towards like actually answering this research question or not? So I just wanted to get Riffin's thoughts on, or the whole panel's thoughts on how they do it.
Fantastic. Yeah, thank you, Mohammed. And can you just say where you're dialing in from and which company or
Yeah,
organization?
I'm with Netrius. I'm actually dialing in from Washington, D.C.
Okay, great. Thanks for joining us. Great question. Lauren, do you want to take that one first?
Sure. Yeah, it's an age-old problem, honestly, for any platform, because when we go into a company to sell Riffin, we have to provide ROI, and so it's a very value-driven question. It can really depend on what's important to a research organization, and so like if you are producing strains for a given program, perhaps it's the length of time that it takes you to produce a strain. Perhaps it's the cost behind doing that, the number of people required to do it. You really have to evaluate different measurement capabilities. Do you find yourself employing extra people to deal with data curation, for example? Is that something you can measure? And so there's a saying that I've stolen from the fitness world, which is that which gets measured gets improved. And I think it's true everywhere. I would simply pick some facet of what you do and start tracking it. And I think the challenge we often run into is we hadn't been measuring it before. And so we won't know when we see an improvement. And so you have to start somewhere and create a baseline.
Great. Ken, how does
I agree
Cargill measure
completely with
productivity?
Well, we have different metrics. So we've got internal metrics where we try to be very careful about capturing specific activities that were improved or indeed throughput improved. But to the point, if you go back and look at development cycle and say getting a new renewable product to market, we never really had a great baseline for that. So I was thinking of the C.S. Lewis quote I saw recently that had to do with not being able to go back and do it again, but you can start where you're from. So it takes some innovative thinking and doing the easy stuff first. I think some of the internal things that have direct measurement, but then I also think it takes some boldness to put a stake in the ground and say, this needs to be to market by this date. You have to timestamp the starting date. A lot of times the starting dates are so nebulous as ideas are discussed, and definitions matter in that regard. But then at some point you can say our goal is to be on the market, and that's going to include capital, putting steel in the ground and those kinds, or you just define the metrics and then see what you can do with that and expect it. We're talking years, and that is part of the problem.
Jessica, go ahead, Lauren.
I was going to invoke Jessica, actually, because I'm thinking of you while I'm answering this. So I'd love to hear what you think about. We could do a whole bunch of measurement, but maybe you could speak to the quality of how we measure these sorts of productivity things. And I've certainly seen cases where we double data output, but that doesn't necessarily mean we're doubling productivity.
Yes, I think that in terms of productivity, whether it's on target for the product goal, getting towards a product concept faster, or is it for building a platform? And in that case, when we think about, in both cases, I think that the quality of the data is, how confident are we that the data are meaningful is important. And then the secondary to segregate by those two targets. In the one case, or in the latter case, we care about keeping negative examples along for the ride. In the former case, we maybe want to down select, and then maybe our learnings from those from those data would be limited to algorithms like Bayesian approaches, probabilistic approaches. Yeah.
Great, thank you, Jessica. We have about 5 minutes left as today's SynBioBeta Live. So if you have questions that you want to ask our panelists, please type them into the box. And also, if we can post the link to the second Zoom room, and that's going to be used for the demo. So if you want a product demo of Riffin, we will wrap up here in 5 minutes and we will jump onto that other room. And if you want to stay around and you have technical questions about the platform, Lauren's agreed to stay on for a few minutes at the end, and he can follow up on anything that was discussed in the panel or ask specific questions that you might have. So we'll be doing that in just a second. We have one question that says, given how complex and bespoke the research process can become, what steps are employed to objectively measure increased productivity, and how long does it take to make those measurements? I guess this is quite similar to the last question, but any further thoughts on that, Lauren?
Yeah, I mean, I think Ken and I said it. It's kind of picking the metrics that are most important and just starting somewhere. There's like a circularity to the answer, unfortunately, which is I can't go back in time and measure things I didn't measure. So I just kind of have to start somewhere.
Yeah. And another question that says, how does Riffin ensure data privacy and security? Does it share or sell data to third parties? And how is it possible that this scenario, could happen?
So our security policy documentation was the very first official document written for Riffin, recognizing that, company and corporate and academic data it's like the most valuable asset anyone's generating. And so no, we would never ever sell any of your data. We take privacy exceedingly seriously. Our core sharing model on the system itself can even protect individuals within a company from one another. And so, things only get shared on a need to know basis, and you have to opt into that. There's no way that it accidentally gets distributed to people who shouldn't see it.
And Lauren, early days of the biopharma industry jumping into software tools, there was just a kind of a no way policy. Would we use a software as a service tool where we don't have it inside of our firewall? Right. Is that now changed? I think you're on AWS and they have integrated security protocols. Do people care about that anymore? Is there such a thing that they want it inside of their company, inside their firewall? Or are they willing to look at
I mean, there's still the conservative hanger-ons, so I'm not going to say everybody's converted, but I will say that not naming names, but a notable pharma company has told us on more than one occasion that they felt that it was actually more secure to move into the cloud than to stay local. So
yeah. Elizabeth, what do you think?
Yeah, John, I think that's absolutely where pharmaceutical companies have gone. So there's been, very high profile recent instances, and I will name Merck since that was public, where on-prem software resulted in, very significant data hacks and data breaches within their organization that was propagated across their entire internal network. And it literally wiped every computer that was connected to their intranet. So I think they, there's a very strong focus on security of software that's deployed in these organizations now. I think it's been neglected for a really long time. And actually, I think SaaS platforms built with gold standard security based on infrastructure like AWS is now seen as the gold standard, and actually on-premise software is really looked at as problematic because of the inability to actually keep those things up to date from a security perspective.
Great. And Jessica, I'm sure you're thinking of the two sites in Massachusetts and California. There's no way you could do on-prem.
Well, actually, when I came to join Bio, we did have on-prem at our Boston site, and I just this one actually makes me chuckle a little bit, because I actually, although I did my training in molecular cell biology and developmental biology, I went into data science via a high-tech company in cybersecurity. And the things that we thought about in terms of cybersecurity, data breaches, and hacks in that space versus the way it's viewed and handled in in R&D and biotech is really, to my observation, very naive. And so outsourcing that to a software company that does have the know-how to think about that is actually a very reasonable approach, in my opinion.
Great, thanks for that insight. We are at time. Thank you everybody for joining us today. If you want to go and get the demo of the Riffin Nexus platform, please click on the link that is in the chat box. Just leaves me to thank Elizabeth Irons, the CEO of Science Exchange, Jessica Louie at Join Bio, Ken Finley at Cargill, Lauren Perelman at Riffin, and also Tim Gardner, the CEO of Riffin. So thank you all. This has been a fascinating discussion. I hope you all stay COVID-free and hunkered down inside your labs doing the great work that you're doing. And thank you all again for joining us today.
Thank you for including me.
Absolutely. So as I said, Lauren's going to stay on this Zoom for a few more minutes. If you have technical questions, then you can type them into the box and Lauren will answer them, or you can also raise your hand and we can bring you on to chat with Lauren. And if you want to go and do the demo of Riffin Nexus, then click on the link in the chat box. And we'll see you again next time on SynBioBeta. We hope that you can join us for the conference, September 1 through 3. It's all digital, some very exciting things that we're doing, including some special VR events where you can run around in VR and meet other people if you have a VR headset. And we're going to be including a lot of interesting sessions that are coming up in the SynBioBeta newsletter. It's a weekly no spam newsletter, so go to synbiobeta.com to sign up if you're not already. We'll see you again next time. Thanks again, everybody. Bye-bye.